There are several types.
All right, we're going to start off this section by watching a short video on DNA expression or gene expression.
The message that DNA carries is passed on to RNA through base pairing. There are several types of RNA. The form of RNA that carries this message is called mRNA or messenger RNA. Because of the similarities between the two molecules, DNA and RNA can base pair with one new twist. Since thymine is absent in the RNA strand, the adenine base in DNA will base pair with uracil in the RNA strand, U with A. The length of mRNA that results from this pairing is called a transcript, and the process that produces the mRNA strand is called transcription. In the strand of mRNA, each group of 3 consecutive bases is called a codon, hence the name triplet code. Each codon codes for one specific amino acid, and the polypeptide chains that eventually result become proteins after appropriate 3-dimensional folding. This process of producing polypeptide chains and thence proteins using the codons carried by the mRNA is called translation. The genetic code provides the relationship between a 3-base sequence and an amino acid. It is important to understand that the genetic code refers to messenger RNA sequences rather than DNA sequences. There are several characteristics of the triplet code that are important. 1. The code is written in linear form using mRNA bases as letters. 2. Each word is composed of 3 letters. A group of 3 letters is called a codon. 3. The code is unambiguous, meaning that a single 3-base code specifies only a single amino acid. 4. The code is degenerate, meaning that one amino acid may be coded for by more than one codon. 5. The code has start and stop codons that initiate and terminate translation. 6. The code is commaless. The bases are read one after another with no punctuation. 7. The code is non-overlapping. Once translation commences, each base is read in order with no overlap. 8. The code is nearly universal. With only minor exceptions, the same coding dictionary is used by all living things. Marshall Nirenberg and Heinrich Mattei were the first to discover the meaning of one triplet codon, UUU. First, they set up 20 different test tubes, each carrying all 20 amino acids. In each one, they labeled only one amino acid radioactively. In the test tube which correctly determined the codon's meaning, the amino acid phenylalanine was the one that was labeled. Next, the scientists added a strand of synthetically manufactured mRNA composed solely of UUU codons. They also added ribosomes and other molecules in a cell-free system necessary for translation of a polypeptide chain. Now they watched translation occur. They expected a polypeptide chain to form with only one type of amino acid present, since the mRNA contained one codon, UUU. In the test tube where phenylalanine was radioactively labeled, they found a radioactively labeled chain of phenylalanine. The scientists knew then that they had a match. UUU codes for phenylalanine. The message that DNA carries is passed on to...
So the last section that we're going to talk about, or well, we've already sort of talked about, is this translation process, and this is going to be done by using transfer RNA, or tRNA. This is going to convert that genetic message carried by mRNA into a particular sequence of amino acids. This is going to recognize both the codon on the mRNA and the amino acids specified by that codon. So this is kind of like a little, I don't know, Google Translate running around and translating every single codon as it comes along and turning it into an amino acid sequence. So this is going to use an enzyme to ensure that the tRNA binds with the correct amino acid, ferries the amino acid to the correct location on the mRNA strand, and then releases it when it matches to its correct anticodon. The anticodon is a 3-base sequence on the tRNA that is complementary to the mRNA codon. It's going to bind to the codon on mRNA, and a specified amino acid is going to be added to the growing peptide chain. Here's just a little example of what that looks like. Here we have our mRNA sequence. It's going to be read by the anticodon that's attached to tRNA. tRNA, this particular tRNA molecule, is always going to have this sequence AAG here, and it is always going to go and pick up the same amino acid phenylalanine. So this tRNA, he only knows two things. This is his anticodon, and this is his amino acid. When he finds a location to bind, and like I said, you can kind of think of this as magnets in a bowl of magnet soup. These tRNAs are going to be floating around. This is going to be our exposed sticky end. When the anticodon finds a codon that it matches to, he's going to drop off or release his amino acid, in this case phenylalanine. To aid in this entire process, we're going to need ribosomes. Ribosomes are going to be our little protein factories. They consist of 2 subunits, each composed of ribosomal RNA and a protein. Their primary function is going to be to bring the tRNA bearing an amino acid close enough to the mRNA to interact. I know I said it's a bunch of magnets in a bowl of magnet soup, but it's going to be a little bit more complicated than that, in the sense that we use the ribosomes, or a spoon, to kind of intentionally push together and make sure that the sequence is happening in the correct order. This is going to allow for the permanent alignment of the anticodon and the codon. So this is what our ribosomal subunits are going to look like. We have a large subunit and a small subunit that this mRNA is going to feed through this groove. This tRNA binding sites are going to occur here, and the protein sequence will be punctured off the top. Here is a table that's going to review the different types of RNA and their various roles in gene expression. But we're going to talk specifically about translation. Translation is going to occur in 3 steps, starting with initiation. This has all got to begin somewhere, sometime. Initiation is going to be controlled by the major players in protein synthesis: mRNA, tRNA, and ribosomal subunits all coming together. Here we have our mRNA with our start sequence AUG. It's going to bind to the small subunit and the large subunit. As soon as it binds into place, our tRNA is going to come in. In this case, UAG carries the start sequence amino acid called methionine. Next tRNA is going to be CCA, which is going to bind to the anticodon GGU and pick up our proline. As we get going, we have to initiate, then we get going. The next step is going to be called elongation. Elongation occurs as the enzymes in the ribosome link the amino acids together. It's going to form a growing chain of amino acids. A polysome is a cluster of ribosomes simultaneously translating the same RNA strand at the same time. So this is a little bit of what that looks like. As an anticodon pairs with a codon, that amino acid is going to be released. So we're going to, here we've got 1, 2, 3, 4 amino acids. This new one is going to come into place. This tRNA is going to release his molecule, his polypeptide chain, attach it to this amino acid here, and now we've got a new peptide bond forming. Now we've got 5 amino acids all linked together, and then we're going to continue this cycle, adding more and more amino acids to this growing polypeptide strand. Here is just a diagram showing that increase of that amino acid chain as we go along. Termination is going to occur when a stop codon moves into the ribosome. Here we have a stop codon, a UAA. This does not have a corresponding anticodon, does not correspond to a polypeptide. So as soon as we read that sequence, the whole thing is going to sort of burst apart. The large subunit is going to move away from the small subunit, the mRNA sequence is going to be released, and the completed protein sequence is also going to be released. And that is it for our section on gene expression. Let me know if you have any questions, comments, or concerns.